welcome everyone to the session 67th of the Beyond Research series that we are organizing with collaboratively with the ACS BioT chapter IIT Delhi and Biopractify and its research division Biolabs by Biopractify. And we have seen 67 past sessions of this initiative which took place as a way to help students network with researchers and you know experts from different aspects of research and industry from all across the globe without any additional free of cost, right? And so this initiative has been really great and has been going on from past 66 sessions, and we have hosted speakers who have been at the forefront of startups, who have been at the forefront of research. Like one of our speaker recently got into Y Combinator as well. So like this has been the spectrum of speakers that we have hosted in this initiative that speaks to the success of this initiative overall. So without further ado, now I would like to introduce our speaker for today's session, which is Miss Parvathy J. So first of all, welcome Miss Parvathy for the 67 Journal Club session. We are really delighted to have you as a guest speaker for today.
Thank you for the opportunity, Vineet.
Yeah, before and thank you. And before starting the session, I would just like to give a brief introduction about our speaker today. So I am really delighted to welcome Miss Parvati for the session today on elucidating putative antimicrobial mechanisms of phytocompounds using molecular dynamics and molecular docking by Miss Parvati from the Department of Biochemistry, IISc Bangalore. So, Miss Parvati completed her BTech in biotechnology from NIT Calicut and is currently pursuing her PhD at the Indian Institute of Science IISc under the Department of Biochemistry. During her undergraduate training, she gained extensive research experience using computational tools such as molecular docking to study mechanistic pathways underlying antimicrobial compounds, drug resistance, and disease mechanisms in pathogens, including Candida albicans. Her current doctoral research focuses on exploring RNA protein interactions and molecular mechanisms that regulate mRNA fat. And today we are delighted to welcome you, Ms. Parvati, here. And this session is also collaboratively organized with the theme of the microbial genomics program that is currently going on. So our students are eager to learn from you, and over to you.
Thank you so much for this amazing opportunity, Vineet. I hope I'm audible to everyone right now.
Yes, you are
And
audible.
yeah. So there is very poor lighting over here. So I wouldn't want to affect the internet that's going on. So I'll try opening my camera and I'll see. I hope I'm visible as well as the slide is moving. And I'll
Yeah,
try sharing my.
you're
Yeah.
visible.
Yeah, thank you. So it's a great pleasure to see everyone around here. So right now in my place, it's evening to overnight. So I'm sure everyone might be having different time zones and timelines. So I thought, so my name is Parvati, and thank you so much for this amazing introduction, Vineet. And as mentioned, I am doing my PhD under the Department of Biochemistry at Indian Institute of Science, Bangalore. So in this talk, I thought I would present something that I have been working on in my BTech, in my undergraduate, along with under the guidance of Dr. Suchitra TV, who was a professor in NIT Calicut, where I did my BTech under. So this was almost a 1 year project that I worked along with Dr. Pooja Shankar Narayanan, her PhD scholar, regarding understanding the various antimicrobial properties on the formulation that they have developed. So I thought I can share one of the work that we have published, as well as the areas that surrounding field that is more interesting and intriguing to work with, since it's more computational in its basis and it requires less experimentation. But still, in the end, with the results that you're getting, if it's statistically significant, you can screen the top ligands and you can move forward for better experimental validation, cutting a lot of costs as well as giving a better understanding based on various simulation studies. So I will be sharing my screen now. So yeah, it's showing that my screen is currently being shared. Do let me know if it's visible.
Yes, it is.
Yeah, sure. So like like shared before, the topic I thought of talking about today is to understand the various antimicrobial mechanisms of phytocompounds via molecular docking and dynamics. So molecular docking and dynamics is a versatile tool where it helps to understand how your small molecule of interest as well as your target protein of interest can interact and bind where on the various pockets and thereby help in its inhibition or in its better functioning. Uh, was there any questions? Okay. So molecular docking and dynamics helps in understanding that particular mechanistic aspects with a computational form, rather than an experimental form, and for various kind of projects and various kind of research, it helps to identify and screen down based on a huge list of data, and it helps to screen down to the best top three or top five without having an experimental investment into this process, but also giving a better top data result. So here in my work, we focused on the phytocompound extract, where we prepared our own phytocompound libraries as our small molecule libraries to screen and perform its various antimicrobial properties based on the different targets that we have taken to understand how the phytocompounds that we have selected would be good combination or a good synergistic effect on various diseases or against various pathogens. So, antimicrobial resistance is a very relevant and it's a very ongoing area of research across various parts of its world, mainly due to its higher death rate and higher I mean, the rate of how much it spreads across different people, as well as the inability of current conventional treatments to treat the diseases, mainly because of the various functionalities or the various, how do I say, the various um, reasons or the various bases on how antimicrobial resistance occurs. It's difficult to treat with the conventional antibiotics and the very conventional drugs to combat antimicrobial resistance. And as you can see here in the charts, antimicrobial resistance death rate is coming on to hand with cancer compared to other diseases and other cases. And as you can see, Asia and Africa are one of the most affected populations because of antimicrobial resistance. And antimicrobial resistance usually happens due to an overload of antibiotics, as which gives the pathogen on where the infection is occurring. It gives them a selection pressure to overcome that antibiotic effect, and thereby, so this can be either via genetic material or mutational changes, giving them a pressure or like various complex biofilm formation to combat the presence of antibiotic, thereby giving a resistant. And this is a major issue because when there is resistance happening in the pathogen and the conventional treatment is not happening, because of the lack of effect of these drugs, some of the pathogens can progress further, which may result in death. When it could have been treated via normal antibiotics. So usually you can see in various hospitals and various clinics and primary healthcare centers, when you go like 5 years before, doctors used to like prescribe antibiotics out of no reason, even though you say like you have a fever, and they'll just give an antibiotic. And the issue that caused because of that is that a lot of people started to get resistant to that antibiotic, like Ziplocs or azithromycin, and because of that they had to be given another stronger antibiotic, and this causes a lot of side effects to the person who's ingesting that antibiotic because of the effect of, because of the strength, and because the dosage that needs to be given based on the various based on the effect of the resistance that's coming, as well as there's also different issues that's happening, and different pathogens that are coming up because of various resistance that's occurring. So usually the most common compounds that used against pathogens or pathogenic infection in general are antibiotics, like I mentioned, that is usually targeted to bacteria, antivirals against viruses, and parasitic agents against parasites, and antifungals for fungi. I hope I'm still audible because there's an external noise that's coming out because of some celebration that's happening. I hope I'm still audible despite of that. If I'm not, please do let me know. Hello? So I'll try to keep continuing on our talks in the interest of time. If there are any questions or any clarification required, just let me know, and I'll be happy to clarify that at the end of the talk. If there's any if you want any emergent questions or urgent questions that you have to ask, I would suggest you can unmute yourself and ask, because I won't be able to see the chat. So another aspect regarding antimicrobial resistance is that in not you can't just think about human health. You need to think of a concept called One Health, where the health of the organisms and the various plants and animals around you is what is responsible for your health. So when you take a good example like bird flu viruses and all, you notice that if you maintain the health of the cattle and the chicken population around your place, that can directly affect the diseases that are being spread from the animals to your to us in the end. So that's why antimicrobial resistance is considered to One Health as this huge area, and people currently are looking and they're trying to understand how we can how we can work with One Health and various organisms and the biodiversity that we're linked with to find it to at least eventually help in combating antimicrobial resistance. And various other compounds and various other chemicals are also used in research. Some have passed the FDA clinical approved trials to also combat antimicrobial resistance as a synergistic compound or as a single compound. So synthetic compounds, which is usually what is given as as an antibiotic or as a compound, are have a lot of disadvantages because of have a lot of disadvantages because of various health effects that's caused by the synthetic compound, that can result in a lot of side effects, which are mainly immune in nature because of the chemical nature of that compound. And it also causes a lot of environmental contamination because of the process it is synthesized, leading to a lot of residues and waste that is being sent off as a slurry or a sludge into our own water bodies. So right now there are a lot of research groups that focus on targeting and trying to find out natural compounds or biologically sourced compounds which are not only environmentally friendly but also more friendly to it also reduces a lot of side effects because it's naturally sourced. It's easier for our body to break it down without having any adverse side effects, but also it's able to perform its function as similar or at least close to the synthetic compounds. Phytocompounds or phytochemicals are one such a group of chemicals that are derived from plant extracts and plant molecules, that has already been known to have a lot of anti-inflammatory, anti-cancer, antioxidant, as well as antimicrobial properties. So phytocompounds, the advantages of working or advantages of understanding phytochemicals or phytocompounds is that it's easily it's more, you can scale it up because of the availability of plants, and you can easily extract them because of already known resources and already known already known strategies and protocols available to extract the various juices. So either you can extract it from the leaf of the plant or the fruit or the root or the stem based on where the compounds are more concentrated in its form. Or you can identify various indigenous plants that are already known to have certain biological effects, but not much research has been there to understand how it has an antimicrobial effect, to see how we can further go downstream to find out its various antimicrobial or potentially even anti-cancer properties, and move forward in the clinical trials of it being a potential target, a drug in curing or at least in reducing the side effects of various cancer diseases. There is a lot of paper, even in the case of COVID, where people have understand and isolated various plant extracts, and they've found out that various phytocompounds or phytochemicals that will be in different collections or in different concentrations in these plant extracts, having a lot of effect against even the COVID virus, which is very interesting. And there were a lot of people who have also tried to understand in a molecular docking and a dynamics perspective on how that various compounds in the phytochemical extract is resulting in the plant is resulting in like inhibiting the various targets of SARS-CoV virus. So when you take phytochemicals in general, they're mainly divided into four categories, such as alkaloids, phenols, organosulfurs, and terpenes. Phenols are more known or well known or well worked with because of it's versatile in against various other diseases, cancers, neurodegenerative diseases, and all. So you'll find a lot of papers where they've tried to use and they try to find out how phenolic compounds have been used in improving various neurodegenerative diseases, as well as it's showing various antimicrobial properties, mostly bacterial and fungal. Alkaloids are also one more thing, and terpenes are also one more compound that you see. But when you take a plant extract in general, you see how these are the main categories that are seen in greater composition, other than various other chemical moieties as well. So in my work, what we have done is we have isolated and created an essential oil formulation from our selected plant extract, in which we have created our own small molecule library via isolating the plant extract and performing GCMS analysis, and then we have performed various molecular docking in silico studies to understand how our various phytocompounds in our data is having different antimicrobial properties by taking different targets against different known pathogenic microbes in our relevant data of interest, which I'll be explaining further. As well as we have performed various experimental MIC as well as agar studies to understand how experimentally our essential oil formulation is having an effect against the different pathogens selected. So there are like various papers that I've mentioned, like I've told before, on how phytocompounds in general are a good alternative or a better alternative in compared to synthetic compounds in against different pathogens and as well as different things. And our paper is one such new paper that tries to understand how one particular essential oil formulation from citronella is being which has already been known in ancient times of its various cosmetic properties. We try to understand how the essential oil formulation contains different components, and that is why it's able to have an antimicrobial effect against various known pathogens. So in our work, why we have chosen to in this order or this perspective is that when you take so our lab mainly focuses on wound healing and understanding how we can create various bioprospective compounds or chemicals to combat the antimicrobial the infections that's associated with wound healing. So when you have a normal wound that happens either let's say as an example, um, it can be a burn wound, it can be a slash wound, or it can be a deep wound. But when you take normal surface wound in general, you can see that our body already has a known immune response that helps it to close and clot it so that the blood does not go further. And after that happens, the dried skin cells acts as a barrier so that infections does not come inside, while the blood cells will have while the normal blood supply that's close to this area of injury will allow allow it to build and regenerate the skins that were lost, and almost it will come back to normal. But in certain cases, there are issues where your wound that was already present here, there might be certain infections that come in along a different period of time, and the presence of this infection can make our wound infectious, and it can cause a lot of side effects and impact based on the organism that's coming inside. And it is a session of importance when the infections that is already been inside, if it is resistant to the already known conventional treatments and drugs, it's going to be a very difficult task, because either you'll have to go for a higher dosage of drug, and even if that doesn't work, based on the infection that has already entered your blood, it can become very fatal to the person. There are a lot of cases where people, when you take about Candida albicans, which is an organism that I have worked extensively during my BTech, which was almost a 2-year work that I had with another professor, Dr. Kabir. Where we found out that a lot of people who are mostly immunocompromised, when their immune response is not working, there are like a lot of cases where Candida albicans, this organism that is already present as an opportunistic pathogen, when your immune response is switched down, or like it is down because of certain infections like SARS-CoV virus or HIV and AIDS, there are cases where this organism that was like controlled or kept in a neutral form because of by your immune response, in its absence, it kind of hijacks your body, and it causes a lot of issues, it causes a lot of diseases, and there are cases it may lead to death because you're not able to control the infection. And there are like a lot of organisms just like Candida in bacteria as well, where it's able to resist, and it's able to overcome the stress that's provided by the antibiotic or the antifungal drug that's been provided, and it makes it difficult. So if you already have, or if you're already encountering with a pathogen that is having antimicrobial resistance, it's important to understand it's important to find out how we can target the protein that is there conserved in the pathogen, so that the pathogen cannot mutate itself to have another variation of that protein. And so when we find out putative ligands or putative lead compounds to inhibit that protein, despite despite it being a resistant strain of the pathogens, since the target protein is conserved, and inhibiting the protein would make the pathogen more susceptible to the drug that you're giving. So either you give it as a synergistic combination along with the already provided antimicrobial, or you provide it as an alternative feature or an alternative function, where it's able to combat the pathogenic the pathogen that's already present in your blood or wherever the infection is present. So usually plant extracts, in our case, like I mentioned, we have created an essential oil formulation of our extract, which is citronella oil, where we have tried to find out how the various phytocompounds that's present in there is able to combat the known pathogens that's associated with wound healing. In our case, we have taken Candida, E. coli, Klebsiella, Staphylococcus, Pseudomonas, and also Acinetobacter. So due to the, even though our work is published, since it's not under open access, we can't show the images that is there in the file. But since our paper will be there, since I've provided it as a reference, I hope based on your institution access, if you're able to access it and read through it, I'll be more than happy to have any questions or clarifications you would have. But I will, because of this, I won't be able to show any images that has been there in my publication because of copyright issues. But I'll try to explain and mention the workflow of what has been done, as well as the already published and already accessible versions of various research that's similar to this, in case you're interested to know more about this area. So this is one such research that I found, a research paper that I have found, in which they have tried to identify various drug delivery systems in understanding how wound healing can be better done with reduced infections based on the various essential oil formulations that they have created from different species, like I've shown here. So in our work, what we have done is that we curated a phytocompound library based on the essential oil formulations that were again like subjected to GCMS analysis to understand the various compounds and the compositions of essential oil formulations that was prepared, and then we have taken those as we have selected those as our small molecule library, and then we subjected it to protein molecular docking. So here we perform site specific docking, where we have identified the top 30 protein targets across 5 different organisms. So this was a docking intensive work where we had almost 150 protein targets that were docked against 10 to 12 different essential oil based phytocompounds that were selected, and from there we have found the top 10 list. We have also we did two analysis. One was to find out where our essential oil formulation was showing a better activity against was whether amongst the top 10 we trying to find out whether the our phytocompound list was showing a better activity towards a particular organism, and two, we try to find out amongst the best amongst the top protein targets across the 5 organisms. We try to find out. We then again subjected the top ligand with our top protein amongst the 150 into 11 combinations of our protein and target docking complexes. We took that and then we performed the molecular dynamics studies to understand how our small molecule lead is acting as an inhibitor, or how it's interacting with our top protein of interest. So in our docking process, what we did was we first identified our protein. We identified the top relevant proteins that are associated with antimicrobial resistance in our different pathogens. We then selected the protein targets, as in the best resolved structure of the protein that's available in database. Here we have used RCSB-PDB as our main database, and then we also took into account the reference inhibitor that was already present in the resolved structure, and mostly we have taken those complexes where our and most of the times our phytocompounds that we have screened had a better score than the reference inhibitor that was already present in the target, showing that the phytocompound list that we had a better effect in comparison to the inhibitor that's already present and is already known in literature. So this is the workflow of how we had performed docking in preparing and curating a protein list. And then for our ligands, what we did is, from the curation of the small molecule library, we subjected them into an ADME screening to understand the toxicity and as well as various absorption, distribution, metabolism parameters, and we took those from the GCMS list. We took those best 10 11 leads which passed the ADME screening, along with so we had a cutoff in GCMS where we took so we had two filters. One was that it has to pass the ADME screening, and another was that we ensured that whatever passed our ADME screening was also in the top percentage of our GCMS, as in like whatever was high in percentage was also what we have taken further for our docking studies. And then we have taken the small molecule leads with a proper 3D STF structure, which is taken for docking. So for our work, we have performed autodock ADT tools to perform the studies, in which we have used site-specific docking, and we have also run it in various in a higher run number, so that the best binding energy that we're getting is also the score that we're getting across different runs. And post our docking, based on the results that we have gotten, post docking, we have also we pursued the top protein with our top ligand, where here we had in our list we had mainly our phytocompound list was acting very generously towards various fungal as well as various bacterial targets that we've taken. So, and even the top protein in the list was also another fungal target that we had. And here are the fungi. One of the fungal pathogens that we have used was Candida albicans. So in dynamics we tried to find out how our top ligand is interacting, or how our top ligand is kind of binding with our protein of interest, the top protein of interest, to understand how the a small molecule lead is a better inhibitor of the top target that we've taken. So amongst the various molecular dynamic simulation studies that we performed, RMSD was done to understand how the structure was changing. RG was done to understand how the various RG and RMS was also done to understand the various structural properties of the protein was changing with respect to in the presence of ligand as well as in the absence of ligand. And RG radius of gyration usually explains how well your protein is stable in the presence or absence of ligand. And we found out based on these various simulations, we were able to find out that not only was our protein complex protein more stable in the presence of ligand, but also based on various other work such as DCCM, SASA, PCA, and FEL. So DCCM helps to identify amino acid residue interactions and how those interaction values are changing in the presence and absence of ligand. SASA is kind of a surface area study test where you try to see how the surface area or the hydrophobic pockets are changing in the presence and absence of ligand, to see if the ligand in the presence of ligand is the protein flipping its hydrophobic pockets, thereby becoming more unstable, for a better lack of terms. PCA and FEL is also another energy based study that you do to understand how the energy levels are changing in the presence and absence of presence and absence of our top ligand. And we were able to show that compared to the normal structure of the protein that's left alone without in the absence of any ligand, our top ligand was able to not only stabilize our protein of interest, but also it was it the our ligand of interest was able to hold on to up to like across like 100 nanoseconds, showing that it is more stable. It is interacting more, and it's interacting well with the protein. Because the issue with molecular docking is that you're only getting one frame of the protein structure, and you try to find out how one, based on whether you do flexible docking or rigid docking, you try to find out how in one particular pose of your protein, how which is the best conformation of your ligand binding to the protein. So it's kind of a static thing that you see. But in molecular dynamics, you're able to simulate both the protein as well as the ligand. So you're able to find out whether the protein is interacting with the ligand across a period of time, and the various properties of and how the various properties of the proteins are changing across the different periods of time. So, and we were able to get a good data as well. And after that, we were able to conclude in our work that based on our dynamic simulations, as well as we did various experimental validations, like we mentioned before. Although our top priority was the molecular docking and dynamic studies, we also did FTIR analysis on our phytocompound library to confirm the various functional groups that's present in our phytocompounds, as well as GCMS analysis apart from the GCMS analysis. And we also and from the essential oil formulations, we also did a validation of our work based on the various pathogenic compounds that's present. Like I mentioned, we have taken 6 organisms for our work to confirm that okay, the molecular dynamic simulations that we have is confirming that our essential oil formulation is showing a better antifungal effect along with an antibacterial effect and an antioxidant effect. So this is our entire process, and this is our entire workflow of how we have done our work, and how we were able to conclude that with the main molecular docking and dynamics work about how our phytocompound list, how our essential oil formulation was able to create a good antimicrobial activity against the various pathogens that we taken. So since here we have gotten really good scores above above almost -7, that across almost across the different pathogens that we've taken, that helps. So it kind of also, in a way, it also means that in that sense, in if you perform if you this topical essential oil formulation, if prepared further as a topical ointment, could be a better it could be a better it could be a good alternative for wound healing infections associated with wound healing, uh, even if the pathogens are resistant in nature in the first place. So if anyone is interested further to understand more about various plant extract based antimicrobial studies, as well as to screen various phytocompounds in your plant extract, and curate your own small molecule library, you can feel free to read all these papers along with the papers that I've mentioned before. As always, you can also feel free to read my published publication along with my co-authors, Dr. Pooja and Ari Devi, as well as Dr. Sujitra, the professor in NIT Calicut. In case you're interested to know more, if your institution provides access to the journal that we've published. And as always, and there are also in case and there's also one more paper that I've given in the previous slides, where there is a database called phytochem where people have curated the list of various phytocompounds that's present and has been published in papers. So it's a combination of text mining as well as people that have already in like inserted or like added in that uploaded in that database, where even if you don't have a small molecule library, a list of your own from various experimental data, that could also be a starting. Those kind of databases like those can be another starting point where you can help in identifying as well as like shortlisting various natural compounds for your studies as well. So this ends my talk. I understand it was a very brief talk due to because of the fact that I wasn't able to put any of the images that I've done in my work. But I hope you were able to understand and you were able to take home some important points regarding the topics that I've mentioned. And if there are any questions or any clarifications required, please feel free to ask. Thank you all for your attention. So I'll stop sharing my screen now. If there is any questions, please do let me know.
Hello, ma'am. I have a few questions, if you may ask.
Yeah, sure.
How do you exactly find the protein that gives the antimicrobial resistance?
Yeah, so this is a huge it's more of a literature review process rather than a hardcore thing. So you can do two things. Either there are databases like RCSB helps. There are a lot of tags. The most of the proteins are tagged in either like protein that is involved in antimicrobial resistance in the database itself. So if you search like so the thing is, there is okay. I'll say 3 points. First is that if you search against the organism itself, there will be like top layer proteins that come across the list, in which you can select and in which most of the proteins will have their own main literature associated with the proteins that's available in the database of RCSP, wherein you're able to find out how relevant the protein is and how you're able to take it and how good of it can you is it to take further for molecular docking and dynamic studies. Another is this huge body of literature that you'll have to go through and you'll have to skim through, where you'll have to find out what is the various mechanism as to how your pathogen is creating antimicrobial resistance, and it differs from pathogen to pathogen, because in Canada it will be more of efflux pumps and various transporters that's present that is giving antimicrobial resistance, but in bacteria it's a different way. They will have various enzymes and they will have various conserved proteins that can either cleave the antibiotic or cleave the already known antibiotic that's given against these pathogens, or it's very conserved, or the protein is so conserved that if you inhibit that, the pathogen can't live. So you're taking all these three things into account. You will get to make your own list of relevant protein targets against the various pathogens, and then you're able to find out and then with this you're able to move forward to the next phase of your experiment based on your requirement. I hope I've answered your question, Thanuvi.
Yes, ma'am. So
Yeah.
whatever the proteins, they're already published already or in literature already.
Yeah. So
Okay.
most of the
I have a
times the
second
protein.
question.
Yeah, sure.
What exactly is the reference inhibitor that you were referring to
Yeah.
in those
Okay.
slides?
All right. So I'm not sure if I'm able to see. I'm not able to if I'm able to show or not. But when you go to RCSB PDB, there are the protein. The resolved structure of the protein is usually bound to a particular chemical compound that is there that's present. So most of the time, the chemical compound that's present here would be already a known inhibitor against this protein. So what some people do is they take the most of the times this inhibitor might be an already known antibiotic or already known inhibitor against this protein that taking. So what some people do is, or and what we have also done, we have not noted in our work, but what we have also done is that we have taken so we take that particular compound in that protein, and then we try to see what is the binding affinity of that ligand, because that is the known antibiotic or that is a known inhibitor to that protein of interest. So what you do is you try to find how what you try to find out what is the score, or on how better is your reference ligand bind, is the reference inhibitor that has been already found in the database binding to your protein of interest. So that will kind of be a reference in as to what are the various essential amino acids that's associated with the protein, what is the a basal line of docking score that's present, that the already known inhibitor is binding and interacting with the protein. So you try to ensure that whatever is your small molecule library, when you filter it down, you try to ensure that your top ligand or your top leads would have a better interaction in the same pocket if possible, and if preferable in the same pocket of where your reference inhibitor has inhibited to, because that would most likely be the essential amino acid that is important or vital for your protein to bind to. And as well as it also helps in understanding if your phytocompound was able to have a better interaction, as in a better binding affinity or a binding energy in while interacting with the protein. So that's why we also take the reference ligand, and we try to find out, okay, if is our ligand able to bind better, is our ligand having a good score, is our ligand having a good interaction with the functional amino acid residues. So that's why having a reference like that would be a good thing. Otherwise, how will you ensure, okay, you have a good score, but what is the basis that you're comparing it with? That's why it's good to have this thing.
Okay, ma'am.
Yeah.
Understood. Thank you so much.
Yeah, happy to help. Someone in the chat has asked a link for my published paper. I'll be putting it in a few minutes. By the time, if anyone has any questions, feel free to answer. Feel free to ask, and I'll do my best to answer.
Yes, Ishita, the recording of this would be also posted. And in the meantime, participants, you can also unmute yourself and even, write the questions in chat box if there are any. I think, Dyuti, you had a question. If you want, you can ask the question, now.
Hi everyone. I'm sorry for the technical issue that happened in between. I got cut off of the team meeting and I've rejoined again. If anyone had any questions, please do let me know. Okay, I'll try answering the questions that's present in the charts already. Kriti, I'll put the link of my paper in a minute. My browser is just loading now. I think there is some network issue from my side. I apologize for this technical error that happened. Uh, Prabhakaran asked, in molecular docking, how will we select the compound based on scoring? So, Prabhakaran, are you asking about compound as in the small molecule, a small molecule, or the ligand, or are you talking about how of the complex, the protein and ligand complex together? If you can clarify that, I'll give my answer accordingly. Sanjana's question was, is it beneficial to learn molecular docking from now? Well, Sanjana, it depends on what is your research interest. So molecular docking is a technique. In short answer, molecular docking is a technique similar to PCR, similar to Western. So if you're asking me, is it beneficial to learn PCR? Prabhagan, I'll come back to your answer. I'll come back to your question in a minute. So if you're asking me, is it beneficial to learn PCR? It depends on what is my research area and what am I trying to answer, what am I trying to work on. So if your research area or if your research question is trying to address how a protein or how a target, as in how a target and a lead, how is it interacting, a molecular docking is a good technique and how you can do that. So it helps to solve one it is one technique which helps to solve an answer to that particular question. That is what it does. So in that way, if you think that this is what is an area that you'd like to work on and this is what you'd like to pursue, learning molecular docking helps to understand that aspect, and that is very and that makes helps to understand various other um, biophysical interactions that's happening between the protein and ligand that is till now difficult and very challenging to perform experimentally. So molecular docking helps to bridge that gap. Molecular docking and dynamics, not just docking and dynamics in that way helps to bridge that understanding which is not able to yet it's not able to easily perform compared to computational work. So Prabhakaran's question was based how will we select the protein ligand complex based on the scoring. So IR in short terms, the more negative the binding affinity is, the better your complex would be, because it means that you would require that much energy to break apart the protein and ligands. So it shows how stable and how good the ligand is interacting with our protein. Ideally, again it changes from software to software because different docking platforms is have different software score cutoffs. If you're using something like Autodoc Veena, usually you'll find papers and publications where they've taken good scores greater than -9. If you go for Autodoc softwares, you'll see like my paper, you'll see like we have taken scores from -7. And if you go for Glide and if you go for Schrodinger, they have a different scoring system. So ideally you try to take the compound that is more negative because it means more stable. But to to go forward with molecular dynamic studies, you need to take into account the number of interactions that's also present, because the number of interactions shows how stable your protein is with your ligand. And it is and so it also tells that if your protein is interacting to your ligand in multiple let's say multiple interactions or multiple bonds, such as conventional hydrogen bond um, or like let's say um, alkyl alkyl bond, a pi alkyl bond, it helps to it's an indicator that when you perform molecular dynamics, it shows that okay, across that that the since there is already so many interactions present, it would indicate that okay, your protein, your ligand is going to be stable because it has that many interactions in this particular static frame itself. It's going to have it's going to have a good interaction in the in a dynamic simulation mode as well. um, Ashna's question was, what other docking platforms is beneficial to understand protein to protein ligand, to understand protein to protein ligand interaction. So Ashna, again, it depends. And it depends in the sense like there are like certain softwares that different softwares have different algorithms. That's one thing. And if you're asking which algorithm is good or good for you to a protein ligand interaction, yeah, you're welcome, Prabhakaran. So if you're asking me that, it depends on what is that you need, because each software has its own pros and cons. So there are like very good softwares like Schrodinger and all that that gives a more elaborative understanding on how your protein is interacting to your ligand, what are the various aspects to these interactions. It gives various parameters, just almost close to which is almost better. That because those are different features that's available in the software itself. But the disadvantage is that it is not an open source software. You need to pay for the license, which is highly expensive, and so you need to be in an institute that supports and provides access to that software. There will be like open source software such as Autodoc Vina. There will be like Glide and some other soft Pyrex and all, which has its own pros and cons. Like Autodoc is a good tool because its algorithm helps to understand how accurate and the binding affinity. It has it has a better accuracy. Autodoc Vina uses less computational power compared to Autodoc, so it's more faster. Pyrex is good for complex or like multi-docking technique, multi-docking experiments, such as where you have multiple docking against multiple ligands, and you need to perform it in a very fast-paced manner. Pyrex has this option where you can do docking all together, rather than doing one protein against one ligand at the same time. So there is that, but there are a lot of cases where people don't accept the scores from Pyrex, and because they prefer the scores from Autodoc tools or Autodoc software because it's considered to be more accurate. So every platform has its own plus pros and cons, and it again boils down to what you would require and what is the pros that you would want in it, and what is more acceptable in your project, and that's how you move forward. I have in the chat, I have added my published paper. So if it is accessible through your institution, I would suggest that you can try to read through it. And if you have any other questions, you can just let me know.